On our last video, we talked about innate immunity in length. This time on this video, we will shift our focus to the adaptive branch of the immune system. And let's start with something that we are already familiar with. You might remember this listing of the two branches of the immune system from our earlier discussion. And of course, this time our focus will be on the adaptive branch. So let's discuss it in a little more detail. However, before we get started, I need to again give a warning. This is a huge topic, and there is no way that we could cover this in a sufficient detail that I feel that is needed to really understand it, so we will just do our best to have a general overview of it. That sounds like a plan to me. Sometimes this branch of immune system is described as acquired immunity, and I think that it is a rather good term to describe it. So the idea here is that an individual gets infected once and then builds an immune memory, which we call as immunity in layman's term, so that if this individual meets the pathogen again, there is already a plan ready how to handle this pathogen. This system is made of highly specialized cells and processes. So here is a really nice diagram showing the innate immunity on the left and adaptive immunity on the right. And different cells that are involved in each. Notice that there is overlap between the two, just like I said in our very first video when I introduced you to the immune system. But at large, if we are having to characterize or compare and contrast each in some way, one could say that innate immunity is more general, so it is not as specific, but it acts much faster. While adaptive immunity is specific, so really hitting the target with an appropriate response. However, it is slower. So, in our last video, we discussed how innate immunity had all these lines of defense, making physical barriers like skin and mucous membrane, chemical protective layers like pH, sweat, and oil, and then of course cell response. This time, we shift our focus on the adaptive branch. So let's see what we can explore with this branch. So as we have seen already, the adaptive immune system branch is very specific defensive system, which can eliminate almost any pathogen or abnormal cell in the body. It does this by amplifying the immune response and activating the complement. However, the shortcoming of this branch is that this system must be primed. This means that we need to have had an initial exposure to a specific foreign substance before this system becomes effective in responding to it. And this priming, as we call it, takes time. And there are many different ways how one may become primed to a specific pathogen, and we will discuss them in more detail later. But some examples would be, of course, having an infection or vaccination where we are exposed to a small dose or inactivated form of the pathogen. Or if we have a baby, it can receive mother's antibodies, for example, from breast milk or earlier through umbilical cord. So, what are the benefits of this system? Well, it is very specific, so it recognizes the target and acts on it. It is systemic, and by this I mean that it is not limited to the initial site. And finally, it generates a memory. This means that if we get a second exposure to the same pathogen, there is a very strong attack against it, as the antigen is already known to us. So, at this point, I would normally play or sing Memories by Barbra Streisand, but in the interest of not making this uncomfortable for you and for me not getting into trouble for using her song, I will skip that. Just know that the memory aspect of this is very important in this branch. And we can divide adaptive immunity to two further branches. Humoral branch uses antibodies which circulate free in the body fluids. When they find an invader, they bind to it. And this is where B cells are going to be important for us. Cell-mediated branch acts through lymphocytes, the white blood cells in the lymphatic system. It can act either directly, which refers to killing the infected cell, or by calling other lymphocytes to deal with the pathogen. So we can think of this as if it would tag the pathogen with a sign, "Kill me." Now we look at T cells. I have provided this rather nice review of these two in your learning system for you to study in a more detail. But now I think it is important for us to familiarize ourselves with some terminology. This is largely just revision to start with. Antigens were foreign pathogens that enter body. They should not be there, they do not belong there, and they are likely to cause trouble if they get to spread and multiply in numbers within the body. So essentially, antigens are the target of our immune response. They are something foreign which we must get rid of. Antibodies instead are the workhorse of the immune system. They circulate within the body, and when they find a pathogen that they should not be there, they recognize it and possibly may also act on it. And often you might hear these being referred as immunoglobulins, IG for short. And some of our testing relies on seeing if we have formed these immunoglobulins against something because we have faced this pathogen. If you wish to consider this visually, I have provided an imaginary model of each here. Notice how antigen, the pathogen in this case, so what should not be in the body is yellow. And the antibodies, they are what protects your body circulating within, trying to detect if there is something foreign. These are green. And it is like a lock and key mechanism. The antigen and antibody must be a match. Your textbook shows that antibodies are always this Y shape. But please note that this is not always the case. In reality, we have many other shapes. Now, I would like to shift our focus on something different. Let's discuss about three important cell types that we see in the adaptive immune system. So we have our lymphocytes. These are B cells and T cells. Remember, these were associated either with humoral immunity, and this was the B cells, or cellular immunity, this was the T cells. In addition, we have antigen-presenting cells. These are often abbreviated as APCs. They play an assistant role showing an antigen to other immune system cells. We will start by looking at B and T cells. And what we are going to do next is to describe the development, maturation, and activation of the T and B cells. So essentially, we are looking at their entire life cycle. Note that both B and T cells share the same steps, so you only need to learn it once. I know that this might seem a little theoretical, but it is important for us to know this process. Your textbook offers some nice illustrations that you may wish to use to complement this discussion. In the last edition that I checked, it was figure 21.8, but that may have changed. So let's talk about what happens. Initially, both B and T cells precursors originate in the red bone marrow. And B and T cells mature in the primary lymphoid organs, and this is bone marrow for B cells and thymus for T cells. During this time, these cells achieve something that is called as immunocompetence. This refers to the lymphocyte being able to recognize only one specific type of antigen. Remember, there were many different kinds, and the antibodies need to match these exactly, like a lock and key mechanism. So when B and T cells have matured, they now only display one type of receptors for antigens on their surface. They can only bind to that kind of antibody. Another important thing that happens is that these cells gain self-tolerance. This means that they are unresponsive to one's own antibodies. So your B and T cells are not going to attack you. Only foreign invader is deemed as a pathogen. That makes sense. This is quite important, right? This maturation process includes quite a few steps during which the cells that fail to have immunocompetence or self-tolerance are deleted. And those who are very interested in this topic, I would not expect everyone to memorize this part. I could share that we actually know the maturation process of T cells pretty well, but it is somewhat less understood when it comes to the B cells. I guess that the take-home message here is that the B and T cells go through an education during which we make sure that they mature to what we and them to be. And if they fail to be that, they get eliminated. So it's like an internal quality control almost. Okay, what happens next? Well, now these B and T cells are ready to travel to secondary lymphoid organs. Remember these from our last chapter. So any B and T cells that are not yet exposed to an antigen are called native. So as these B and T cells travel from the primary to secondary lymphoid organs, within the circulation, there is a chance of an encounter with an antigen. Well, if this encounter happens, this causes the T or B cell to continue its development. And this would include the cells multiplying in numbers and becoming activated. And this means that they have two plausible futures. They can either act on an invader. This would be what would cause an infection. Or they can become memory cells. These cells are important as they stick around in the immune system in case the same antigen is met again later on, so that the immune system is able to act quickly at that time. This is what we often think of becoming primed or prepared to a pathogen. We have a template against it based on our previous encounter. In layman's terms, this is what we think of when we talk about immunity, right? So that's the life cycle of B and T cells. And this is all I need you to know about B and T cells at this level. So that was our B and T cells. Now we should look at the antigen-presenting cells. They do exactly what the name suggests. When APC meets an antigen, it cannot act on it itself, but alerts rest of the immune system, almost like, hey, this is an antigen here. So they are basically helpers. And they are different types of antigen. There are different types of antigen-presenting cells: dendritic, macrophages, and B cells. I will leave covering those for your own interest based on the textbook. So, we have seen this slide before. If we wish to recap, humoral immunity, which is shown on the right, was all about being caused by the antibodies. And in cell-mediated immunity, which is shown on the left, There was no antibody involved, but this is activated in response to an antigen. You will have this diagram as a handout in your learning management system, so feel free to check it out from there. Now, I think that we will wrap up this video here. In our last video, which you can find after this, we will talk about primary and secondary immune responses and also some more about relevant clinical relevance. This is a hot topic now, I know, so maybe it will help to explain that a little too. I will see you in that video.